Good evening. So I'm going to have my talk divided into two sessions. First, I'm going to talk a little bit about genomics because that's what I do. And most importantly, I want to talk about genomics because genomics is absolutely at the basis of cancer, because all cancers are actually caused by a defect in the genes. And I also want, because right now I'm sure you know the precision medicine being practiced here in India also, and the precision medicine era was started with my group in 2005, sorry. So, and the later part, I think most of the time I will be spending about what I have done with the BVGs can help. I'm calling that as a compound 7, and I will talk about that more as we go. So, as you know that this is Bill Clinton and Francis Collins. So, US government realized everything in our body, everything in our life is actually controlled by genes, including the development. The set of the genes which are inherited from the mother and father actually knows how to develop the body and how to regulate each and every function in the body. So basically, all the secrets about our body are present in the genes, and this is why US Congress decided to sequence the first human genome. So we will know the secrets of the human body. And President Clinton actually announced this in 1990, and this project was started. It took like more than a decade, and there were so many countries involved. Obviously, India didn't participate in that. And at that time, the cost of sequencing first human genome was more than $3 billion, took 10 years. Today, if anybody was interested in sequencing their genome, I can sequence it for $300 in less than 2 days. So that's a technological advancement we are talking about. And that has made possible the precision medicine. So this is the first study so when human genome was completed in 2003, everybody was very disappointed. And one of the reasons was that nothing secret came out. So basically, people thought that there are 100,000 genes. So actually, there are only 25,000 genes in human body. I mean, I don't want to go in detail, but there are so many myths. So people think that right now there is so much discrimination, Indian, Chinese, even in Maharashtra, in my hometown, Muslim, Hindu, Brahmin, Jain. There's so much discrimination, but people don't realize 99.9% we are genetically identical. There is only 0.1% variability which makes us who we are. And that is the fundamental basis of the precision medicine. My group published, I should always say it's my group, it's not me alone, published this paper in 2005. So 2 years after the completion of the human genome, which made everybody, Congress was saying, why did we blow up $3 billion? It's waste of money, till this paper was published. Because we utilized the information which was provided by the Human Genome Project to identify the gene for the macular degeneration. And now I think we know complement factor H is the gene which is responsible, and we know the mutation, and now we can then, once we know what is causing the disease, then it's easy, or there is a way you can develop the medicine. So this publication started the genomic era and the precision medicine era. So all the credit for current genomic era and precision medicine is actually given to this publication. This made on the cover of Science. So since then, the genomics has become such a hot. As you can see here, there are so many news articles, and I guess my animation is not working here. But now I think, you know, newborn screening is very popular things in the United States. Every baby which is born actually is screened usually by proteomics technology for 60 genes. And these are the 60 diseases which are identified within first few days of baby's life. The baby's life survives, for example, phenylketonuria. But the proteomics technology has several limitations. So with genomic technology, you can screen the entire genome. And one of my very good friend and from Satara, Jay Shenduri, he developed the technology by which you can take the blood from 3 months pregnant woman and determine the genome. And even before the baby is born, we can actually predict what this baby is going to have the diseases in his life. So basically, this is actually was on the cover of Time magazine again, invention of the year, because everybody is actually using genomics to identify not only the disease, but this is also for to know what can happen or prevention also. So then at that time in 2009, the cost of the human genome was still like $10 million. So it came down significantly. So me and Dr. Richard Lipton, who is now the president of the Rockefeller, so we decided that 85% of the disease-causing mutations are present in the coding region of the genome. So as you know that genome has 3 billion bases, but only 1% or 2% of that, like 30 million, is actually genes. Rest of, at least I used to, when I studied, it used to be called junk, but obviously we are finding out that's not the junk. So basically, we decided why sequence 3 billion bases, spend so much $10 million and generate large amount of data. Is there any way you can just sequence the genes alone, which is just the 30 million bases? So we developed this technology called exome sequencing. This is being used even in India today. So we not only developed this technology, but also use to identify correct the prognosis of one of the Turkish kid. So the kid was actually, they were actually thinking it has Bartter syndrome and was not responding to the treatment. So we quickly sequenced the gene and we corrected the diagnosis, and the baby was able to survive. So this is the first example that we were able to use the next-gen sequencing technology to diagnose or correct the diagnosis, and also develop the technology which brought the cost down to like $100,000, so 10 times less. So at that time also what happened in 2009, President Obama became a president and the economy was actually doing very bad. So he announced the stimulus package and me and my collaborators were able to attract $60 million. And at the same time, I also got a grant to study the psychiatric disorders by National Institute of Health. So the combination of this publication, Obama stimulus and my grant, Yale University asked me to establish the Genome Center. So currently I'm wearing multiple hats. One of the hats is I'm also head of the Genome Center. Quickly, we have $20 million worth of equipment, all the latest equipment, and I still have $10 million in the bank if I need any equipment. University gave us a fantastic space. You can see the lab here. You can see some of the sequencers. We have a very big cluster here. You can see that is $3 million cluster. So the thing which I wanted to point out here is we have been doing, my lab was the first to use NGS, next-gen sequencing, to do CLIA. So since then we have been doing cancer clinical diagnosis as well as other disease clinical diagnosis. So usage of this center has resulted into 4,000 publications, and out of that 400 are high-profile discoveries such as Nature, Science, New England Journal of Medicine. So you can see the impact, and that impact is obviously actually realized in the last sentence here. This center has enabled the university to attract more than $1.5 billion of funding, that just from National Institute of Health. So this has been a great success. So I want to talk only about one publication which we did around 2009, and the reason is this is the publication which realized the importance of the precision medicine. So this is a melanoma case. We sequenced approximately 150 melanoma cases. At that time, there were about 10 to 15% of the cases responded to chemotherapy. The remaining cases didn't respond for chemotherapy, and nobody knew the reason until we did the sequencing. So here we identified this row GTP is called RAC. It's a crystal structure is shown in the switch one. There is a one base pair change from proline 29 to switch is to serine. And this simple change, just one base pair change, made this protein extremely active or uncontrollable. And that's what led to the growth as well as you can see the binding and caused the cancer. So we have many publications after that. So we know that there are at least, so one might think if just melanoma, maybe only one gene is responsible for melanoma, but that's not the case. We identified at least 25 other genes which are responsible for causing melanoma. Obviously, this is answered our question, why only 10% of the cases were responsive to chemotherapy, because they were actually targeting only one or two genes, not remaining. So this is where we realize the importance of precision medicine, because now we have technology by which we can sequence each and every cancer tumor and identify what is causing the disease and then treat it. So what happened over the years, the cost of the sequencing came down significantly. As you can see that this is per bed. So current, so it started with $3 billion, as I mentioned, we can do it 300. So it became affordable now. And also competition cost has significantly come down. My first computer in 2010 was $5 million, now I'm spending only $1 million. So because of this, the time was right for President Obama to announce the precision medicine initiative. So as I mentioned, Dr. Lipton, who is my mentor, now he's president of Rockefeller, he was asked by Obama to implement the precision medicine program in the United States. And the reason, and the cancer was selected as a test because cancer is 100% caused by genetic defect. So this is the beginning of precision medicine I'm talking about. So what is precision medicine? Obviously you know, but I just, for those actually, I want to make it very simple. As you can see that traditional medicine is actually one size fits all, but we know one size of shoes doesn't fit everybody else. We are all individuals. We are all different, with different heights, different injuries, color is different, shape of the nose, color of the eyes, everything is different. So because of that, what is happening is that when person is treated with one medicine, because everybody gets the same medicine, because that's the only medicine we have available, then the effect is not optimal. Some patient will respond, some people will have no effect, but most of the people will have adverse reaction. So now we have a technique by which we can characterize the disease of each person. And then based on what mutation is there, then you can, if there is A mutation, you can give A medicine, so positive. B mutation get B medicine, C mutation gets C mutation. So basically patient receives the personalized treatment based on their genetic cause. Right now we are doing genetic cause. We can also, we'll do proteomics also later on, because proteins are at the end of the day are the one which actually brings out the function in the body. But right now proteomics technologies are not, they're very tedious to work with. So this is a precision medicine effect. And now I just wanted to give you this background of what I have done, and I think I've been fortunate enough to be in a team environment where we were able to contribute so much to the humanity, develop the exome sequencing, identify disease-causing mutations. And I want to make sure somebody might ask me, okay, what's the big deal about you identify the mutation? So, but we don't stop there. We actually do the functional studies, test that mutation in other CRISPR and other animal systems to make sure that is the one which is really causing the disease. So we go way beyond that. So anyway, so now I want to switch my topic and talk about the BVG compound. And one of the reasons I accepted to do this because of my genomics background. So this is a very interesting story. I would like to tell you the history. So I think I still remember the evening when I met Hanumantrao Gaikwad. I think that was probably the first time I ever met him. And he was telling me he has a medicine for almost 100 diseases. And I'm very skeptical, and on top of that, I'm a scientist. So I was just thinking in my mind, who's this guy? Is he God? That he has a cure for almost every disease in the world? I mean, I didn't know him then. Now I know him now, but back then I didn't know him. We are from the same town, but I have left India almost 40 years ago. So basically, I was very skeptical till I met Dr. Pawan Singh. So when I had some discussion with Dr. Pawan Singh, I realized this person is very authentic. He has a credibility, and he was able to convince me to test this compound in my lab. So that's how I ended up getting this compound to test in my lab. And I'm going to contradict Dr. Hanumantrao because he said I spent all the money. That's not true. And the reason is not true. BVG also paid a lot of bills because they were genuinely interested. So it is to their credit that they were really genuinely interested in knowing how this works. So again, Dr. Kundu had done some cell survival study. So initially we thought we should start testing this, start with that. Obviously, I was sent extract of some compounds, and I believe these were bioactive compounds because all the experiment I'm going to tell you are there in tissue culture cells. Obviously, most of the compounds are actually metabolized in liver and they become active, but this was already active compound. And I still don't know the formulation, I never asked. So basically, when I tried to do it, our biggest problem was we couldn't get that into solution. So I thought then again, here you go. And I'm very skeptical about the Ayurvedic herbal supplements, very skeptical because I've seen my family, my mother. And so I should tell you something else. I I discovered the gene for macular degeneration. I already told you that. Unfortunately, 10 years later, I was diagnosed with very severe case of glaucoma. And as you know that 30% of the cases of glaucoma is caused, the cause is not known, because 70% cases are caused by increase in intraocular pressure, but the remaining, and I happen to be in that 30%. So I, obviously with my connection, I went to Harvard, Yale, all top doctors, and so finally I decided to start taking treatment at Johns Hopkins and Columbia. So they put me on a clinical trial. So they gave me very heavy dose of nicotinamide. 3 milligrams. So obviously they say you have to monitor your LFT test and make sure that it's not affecting your liver. And obviously after 2 months there was a adverse effect, so they asked me to stop it. So at that time I asked he's a head of ophthalmology at Columbia, can I try a herbal supplement? And he was excited. Is there a herbal supplement for this? Because so I said yes, there is. I'm working with a company called BVG. He said try it out. So when I came to India, I took this supplement. To my surprise, and this is where my skepticism, of course, which was already vanished when I saw the CAN results, which I'll be sharing with you. My liver function tests were normal. So today, after 3 years, I've been able to, I'm still taking that medicine, and my liver function is still normal. So I don't have much options, so I had to take that. So basically here, you can see that we basically repeated Dr. Kundu's experiment, and then when we talked to his postdoc, he told me try ethanol, and ethanol worked. So we tried it on 3 different cells, the breast cancer cell, colorectal cancer, and we also decided to take primary cells from kidney. These are normal cells. So as you can see in this graph, colon and breast, obviously you can see that the cells actually start dying as early as 1 hour, and I think after 24 hours almost 50% of the cells are dead in both the cases. I think, and you also can see this is dose dependent, because the higher dose actually is killing the cells much more rapidly than the lower dose, and the normal cells are unaffected here. So later on, I think we also decided to see whether we can kill almost 100% of the cells. So we extended the treatment to 48 hours and 72 hours. And as you can see, and we also take DMSO and ethanol. These are the solvents in which we dissolve the compound, and we wanted to make sure they are not the ones which are causing the effect. And in addition to that, we also take positive control. You guys know cisplatin as well as mitomycin C. These are well characterized chemotherapeutic agent. So you can see that there is no effect with DMSO or ethanol, but this is compound 7. This is the BVG compound 7, killing the cell very effectively, and this is dose dependent manner. Actually better than cisplatin and probably similar to mitomycin cell. So 90% of the cell death was achieved after 72 hours of treatment. So we realize it's something real. It's not haphazard that anything you don't just toxic would have killed the cell right away, but this is killing the cell depending on the time and the treatment dose. So next question was, so somebody I'm just telling that cell death can be achieved, and normal cells seems to be unaffected by compound. Another thing also that obviously I had a question, how come one compound actually be effective against all the cancer, but I think once you see the data, you will know the reason for that. So then we decided to find out the mechanism of action, because that's where we were more interested. So we decided to start with RNA-seq experiment to see which transcripts are being actually changed in both treated and untreated cells. So we actually have untreated 2 hours, 4 hours, because we didn't know what, we wanted to do the time course, and as you can see here, so this animation is not working, by the way. So basically, you can see that 2 hours there were 92 changes, 12 changes only, 4 hours only 14 changes, whereas at 24 hours there were significant changes. And this is reflected here in the PCA plot. As you can see, the PCA plot, actually the same samples should group together. So you can see that 2 hour, 4 hour are more or less grouping with the controls are untreated, but whereas all 24-hour time samples are actually grouping together. So later on we decided to work with 24-hour sample, because that's where the effect was more pronounced. So this is a heat map of the gene expression. This is all the genes, differential expression genes we have shown here. So as you can see that this is normal, so control, untreated, 2 hours, 4 hours, and 24 hours. As you can see that there is a transition. So this means the expression is higher, then you can see it's getting lower and lower here. So these are the set of the genes. First group of significantly decreased expression. These are the genes which are the expression levels are changing. When in group 2 there is significant increase in the expression, whereas in group 2 there is a moderate increase in expression. So again, expression changes are occurring gradually over time. As you can see that it's started seeing more clearly in 4 hours. So then what we did, so these are all genes. So obviously we use P value as a cutoff to identify significantly changed genes. There were about 800 genes. And then we use the cake pathway and go pathway to understand what pathways are being affected by this compound. And to our surprise, most of the chemotherapeutic agents really just kill the cell, are toxic to the cell. But unlike here, you can see that. So you can see the proliferation is also affected. You can see the green. Then the migration and invasion is also actually affected, as well as immune response to tumor are also actually changed. So basically, it looks like this compound is acting in multiple levels and in multiple ways, and obviously that's understandable because this is not one compound, this is a mixture of different compounds. So that was good to know. So then we did further digging. We wanted to see cancer-specific genes which are being affected. So this is apoptosis. As you know, that apoptosis is a mechanism by which the cancer cells die, or this compound is killing the cell. And you can see the genes which are upregulated here, the red, and the genes which are downregulated are shown here, that two distinct groups. And I have shown the function of some of these genes which are changed. This is, for example, BAK1 encodes the pro-apoptotic protein, initiate programmed cell death. So you can see the genes which are involved in apoptosis as well as programmed cell death are increased. That means their function is increased. That means it's specifically acting on those genes. Whereas the genes which were downregulated were cell proliferation, vital first cycle, cell growth proliferation. So this is just a list of the genes, and we are doing a lot of other studies, but I just wanted you to get an idea. This is not a hit or miss. This is a real phenomenon because it's affecting the genes. Then we also looked at senescence because this is where the growth is arrested. So you can see again, these are the genes specifically NRAS, senescence, anti-cancer mechanism, cell cycle arrest, survival growth, cell cycle inhibition, all the genes which are really known to be involved or should be involved in cancer progression or cancer death are actually being affected by this compound. Cell cycle also we studied. So we can see a lot of genes, TGF-beta3 and TP53, which is a tumor promoter, right? Because that's a gene, P53, sorry, not TP53. So we can see that the list of the genes which are actually downregulated, upregulated. I can't go in detail considering the time limit, but basically these are all the genes which we would have expected to go up or down with this treatment. Again, cancer. So you know that these are universal like FAS, JAK, FGF receptor, so RO1. So these are the mTOR, June. You can see this, some of them are oncogenes. So all the right genes are being hit. So this now made me clear how come this compound is affecting against all cancers, because this compound is actually affecting not at one gene but multiple levels, and the genes which are actually affected by downstreams, because you know how the pathway works, because there are tens to hundreds of genes which are involved in one pathway. So it's affecting a thing at multiple levels to bring about this action. So then we also wanted to say, as you know, that if one gene is actually affected in a pathway, it affects the other genes. So we decided to see, okay, what are the effect of these genes upstream as well as downstreams. And as you can see that we found total 100 predicted downstream pathways. But if you read the description, they are all cancer and they are all decreased. So this is no coincidence. So then we decided to do ATAC-seq. So what is ATAC-seq? So ATAC-seq is a method which is used in cancer research to map open chromatin, identifying active regulatory DNA elements and enhancer promoters that drive tumor growth, progression, metastasis and everything else. So again, you can see that 24 hours are actually grouped together. So we actually, I'm showing you the list of the genes which are drastically changed. You can see these are changed genes. So we were able to identify 79 and 54 differentially open region genes. So open region chromatin means the gene is active. So whenever gene becomes active, the chromatin unwinds and opens up. And when the gene is not active, the chromatin closes down. So the gene, there is no access for the transcription factors and other factors which start the initiation of the expression. So this is very important technique we did, and again, so we did the cake pathway analysis of these 1700 genes, and you can see that then again so many pathways were identical: leukemia, metric acid. So all these focal adhesion. So these are all important: cyclic MP, proteoglycans in cancer. All these pathways are being actually identified. So again, this also proves that this compound is actually really very effective against cancer. So we also identified, I'm not showing all the data because we think we know the mechanism, how it works, but I just wanted to give some example how it affects. So this is another important gene we found, but this gene is involved in DNA repair and so that's a proofreading function for DNA polymerase. Inflammation, so again I told you this compound also acts on inflammation as well as cytokines and other factors. Toll-like receptors metabolism, controls metabolism. Obviously metabolism is very important in cancer. And this is ELF3 is a transcription factor that regulates the cycle. Then this particular gene again transcription tumor suppression involved in shutting off genes. So you can see the list of the genes which are so important for cancer biology being affected by this compound. So we also wanted to do single cell genomics. So I don't know, in our cell, our body, the biology doesn't happen in tissue. Biology happens in one particular individual cell. So single cell genomics is a very modern techniques and very expensive techniques, and we decided to do it because we wanted to see, because as you know, tumor is clonal. So there is a lot of diversity, and there are a lot of different type of mutation present in one tumor. Out of that, one is driver mutation. Obviously, we are actually working with the tissue culture cells, so we won't expect clonal, but we still wanted to see whether there is any difference in some cells are being affected by one gene versus other genes. And actually, as you can see in my next slide, so all these cells actually are separated based on their expression profile. There are so many, 30, 40 clusters. And so next slide actually tells you actually the important genes which are involved in some of these clusters. For example, this cluster has NEAT1, and this is very important gene because this is sort of act like oncogene. And but here this expression is reduced, so basically oncogene effect is actually reduced. So we can see that this particular one has this gene, these particular cells are this. So we were able to separate the cells from each other based on their expression profile. And these genes are again widely involved in cancers. Some of them act as oncogenes, as I mentioned it to you. So and regulate proliferation and growth. So I mean there's still a lot of work needs to be done. I think I'm short with the manpower because of the funding situation in the last 5-6 years has become extremely hard to hire new people. But I just wanted to summarize so far what we have done. We think that it's working with kappa beta 2. This is a known gene, you know, that is very well involved in cancer, very well characterized in cancer. So most of the chemotherapeutic agents actually are toxic to cell. They kill and they either cross-link with the DNA, they alkylate the DNA and kill the cell. But BVG is different, and you can understand why it is different because most of the chemotherapeutic agents are highly pure, so they will act only on one thing. But the BVG's compound is mixture of different herbal products, so they are different effects. So it not only it exhibits multiple anti-cancer activities, including cell proliferation, migration, invasion, as well as modulation of the immune response. So I think this is very important, modulation of the immune response, because it's just not killing the cells or stopping the cells from spreading or getting metastasized, but it's also activating the immune system, which probably is killing the cells. Which we haven't gone there. We still need to do a lot of work. But so again, as I mentioned here, the work is currently being underway and more in-depth analysis using PAPAM. Now, Dr. Gaikwad's, Hanumanta Gaikwad's daughter has joined. She has a lot of experience in genetics, so I'm hoping she will be able to carry forward our work with me to do this analysis, because most of this can be done in the office by just looking at each and every gene, how it is working in a pathway, how it relates, what does it mean, so we can start understanding the full picture and the full mechanism of this compound. So thank you very much. My last slide is here. I want to thank BVG, particularly Gaikwad sir, because for giving me this opportunity to talk in front of you. I also want to thank Dr. Pawan Singh and Shri David, and now I would like to thank ahead of time to Aditi also and her to be her fiancé also, that hopefully we'll be working together. Obviously, I want to thank my collaborators, my staff university, and I also wanted to thank all these funding agencies who have provided me money to do this research. Thank you very much.
Request anyone, if you have any questions, we are open for it.
Thank you for the excellent talk. But see the amount of hits that this particular compound is doing at various levels, apoptosis, immune system. There has to be a mother pathway which is triggering all this. So are you working towards identifying that? Because this is a very wide variety of effects. So either one compound is hitting a mother pathway causing all this, or there are multiple compounds acting. So what is your take on that?
Yeah, it's a very good question. So as I mentioned before, I don't think it's affecting only on one mother compound. So obviously, so they are actually have separated this mixture of compounds with HPLC. So now is a time to try each and every one of them and identify that compound. So that will be our next. And I have listed all other experiment which we want to do. Other thing we want to also do is we want to take this particular variants, I would say, and test it in biological assay to make sure or confirm that this is exactly causing, because we can do CRISPR. There are so many other functional assays, and we have already started. We have already done some and proved that that's how we know that NF kappa beta kappa 2 is one of the genes which is very basic to do, because that gene is also involved in immunomodulation.
Dr. Mane is a very interesting and a very revolutionary talk, Dr. Kiman. Being a physician from allopathy side and also in Ayurvedic background, very difficult for a modern patient to believe that somebody can come with Ayurvedic medication which can take care of almost all cancers. Obviously, you explained that very elegantly the way we are done it. I being a clinician, I look more from the patient's point of view. Other than what Vinaya asked you, going to the laboratory, finding out the pathway, that is a different matter. But ultimately, the patient is interested in getting the result, and that is most important. So, would you be interested, because when you talk about prostate, being a urologist, and kidneys, my interest had come. So we keep on seeing so many patients. In fact, I was remembering one patient, we had Mr. Chaudhary. Few days back, I mean, in hospital, he has taken everything, hormonal, chemo, radio, including lutetium. This is the most latest of the treatment. When everything has gone and he was so in a bad condition that he just waiting for death to come. So in case we just connect, we try to, because in India it is easy to tell the patient is herbal medication, and there will be much ado about it, I'm sure. So let this take it further. Maybe in few cases also, if you can get the result, I mean, we can see whatever samples you require, we can take the permission, send it to you. We monitor the clinical result. So that is, I think, that is the taste of pudding, I would say, that is most important. If we are agreeing for this, today only I declare that we don't mind working for. This is a big thing for the humanity. And I really congratulate BVG group, my close friend Anurag and of course now you are coming to that group. So let us work together. Whatever lab work can be done at your level, but clinically I think we should connect. Thank you very much.
So as we talk, this is a really good point, because at the end of the day, what matters is patient's life. That's why we are doing all these things, right? So I think now Dr. Jayashree has joined the team. So I think I'm going to start working with her to see a particular patient who has a cancer. So we will collect the sample or biopsy before the cancer or the treatment, and then collect the sample after the treatment to see what changes are actually being made. So we can compare before and after. That's something which we should be able to do without no problem there. And we may not have to do all these studies, because by that time we will know exactly what we need to do, so we can just do that as a litmus test. And so I also should mention that. So I perfectly understand that why many of Indian doctors are not very keen on using Ayurvedic or herbal or phyto medicines. I think one of the reasons is the quality. And if I already have a meningioma, and I know it's not growing for 8 years, and I have another paper in science which I wanted to talk to you, but that because of the time I didn't. But basically, by just knowing the location, we can tell whether it is cancer or benign. So mine one looks like benign. But if at all, I hope I don't have to. But if I have to take, I will take BVG medicine any day because I trust. And I trust because, of course, I now know the man, Hanumantrao Gaikwad, but I also know his team, Pawan Singh, and I'm getting to know now Dr. Jayashree. So basically, I definitely, in fact, one of my friend, very close friend, he was a professor in neurosurgery, just passed away yesterday. He had a pancreatic cancer, and I tried my best to convince him to take this medicine, but I guess it was already too late. And I was planning to take it with me to give it to him, but he already passed away. So I have been referring my own relatives to BVG clinic to take this medicine. So what I would urge the doctors is to just keep the open mind. I'm not saying your mind is not open, because I understand why your concern is. But when the product is so well validated, so well quality controlled, so I think when there is no hope, I think I don't see any reason why not to try this medicine.
All should take it as soon as the cancer is detected, rather than wait for going at the stage 4 and taking the medicine as a last resort. And nothing is working, like Dr. Patankar said, that now everything is not working and we will try. That's the maximum patients we are getting. But we want to get the patients who will take it at the first go.
So he has a
I
question.
would agree and disagree both. See what happens, standard care you cannot deny to the patient, first thing. So you have to give them the standard care which has been proven. What we are trying to collect the data first. So if you say I got a patient of cancer prostate and should I give him? No, because I am offering the standard care. Maybe agreeing with that, agreeing with the next is I said agree and disagree on this. Agree we can give the standard care and see the results. But see, I will give a large number of sample size for me to say that. Unfortunately, surgery I cannot very openly here. So many Ayurvedic doctors they claim they treat the cancer, and we see coming to us in such a bad condition. Then we say why you are going to that particular, and they defend the Ayurveda. See, science is correct. The people who have practiced and defend the science. They don't know the exact way, the way Sir has explained us. That is the way when we can convince ourselves. Otherwise, I am a modern physician saying I have Ayurvedic practice. They said Kapil Vaisa practice karte. So people laugh at us. So thing is, if you wanted to prove, why I am saying you might be a little disheartened. If you see some improvement, that is the first thing which we can tell the patient. I won't say he's going to get cured once you see a patient with meds, I'm sure. But by experience we know the quality of life, lot of things can be done. But that is how one has to go. And maybe as I said, we detect the cancer, we have to do a protocol with Sir's help, then we can come on with some publication and tell the scientific community, yes, it is working. Thank you.
Yeah, I can just say one thing, that when you start treating cancer patients who are stage 4, at that point there is exhaustion of defensive mechanism of the body already. So don't expect anything to really give you miracles at that stage. If you want a drug to be tried, it has to be tried early. You can try it in parallel, you can have two RCTs, you can have two groups, one with the adjuvant, one without adjuvant. Starting late, I think, and then saying the drug doesn't work after the body's defenses have given up. Almost this biological exhaustion. There nothing will work. So we have to start early, if you have to say, before dismissing. So I would urge if you want to start a drug like this or any other drug it has to be early. You can't start it late.
So I know he has some question, but I would like to say, I think I'm pretty sure you all know. So right now I'm doing CFDNA test very routinely. I do it myself every 6 months because when I was doing my PhD, I used a lot of carcinogens. That time we didn't have gloves, ways to do mouth pipetting and all those kind of things. But the CFDNA is going to help us a lot to detect the cancer way before even the first stage. And I think that technology is just improving day by day. When that happens, I think there is one company in India is already doing that. When that happens, I think the detection will be very easy. Okay.
First of all, I would like to congratulate you that you have explained the genome in a very simple and explained manner. So, heartiest congratulations to you as well as Dr. Mr. Hanundrao Gaikwad as he has invented this compound 7. Regarding this, I would like to ask you that can we use this C7 for immunosuppression?
Can we use it for? Can you repeat that, please?
Can we use C7 compound 7 for immunosuppression in plasma?
So I rather use the whole product rather than just compound 7. This is just the fragmented and active form. So because I haven't tested other compound, there are obviously 7 compounds. So
Are you planning to do that?
I am definitely planning. Whenever Gaikwad sir tells me yes, go ahead.
Basically, it acts as an immunomodulator. We have studied other studies, so we have seen we have enough data available. It made this medicine acts as an immunomodulator. So where immunity is low, it increases the immunity. Where immunity is high, it decreases the immunity. So it makes modulation and helps patient to cure faster.
That means this will be very helpful for post-op cases,
Yeah.
which involve a lot of expenses. Then another question is that, have you tried this in actual diabetic macular degeneration?
So what about macular degeneration?
Retinal problem.
Yeah. No, I haven't tried in other people.
You
No,
have tried on
no, no.
yourself or glaucoma?
I thank God I still don't have macular degeneration, but I have glaucoma. So I have told the doctor, but I think these guys are so busy. I think this is something which I'm currently talking with Dr. Pawan Singh, whether he can give me the extract of this liver drug and I test in that. That's very easy to do it.
So that
Because
is
in
also
India, there are
Liver
many
driven
cases of diabetic retinopathy, and that would be
Yeah.
So-
So liver, we have tested our medicine in Dr. Shiv Sir's lab ILBS. And sir, that report is will be completing that report in next 2 months' time. And we found out that it is better than any other allopathic drug and better than very famous liver drug. I don't want to name the company, but it is better than 6 times better. And sir himself has taken that medicine when his liver function was got affected, and we told him that you test it. Sir, he got results in 15 days. 15 days. So he insurance, he said that he was there going to give lecture in Hyderabad AIMS, and he said that I would like to test my liver function test, and the liver function was normal. He didn't believe that this is normal. Then he went to US in Yale University, again tested, and got the same result. So now he said that liver medicine also is working very fabulously.
So I would like to request you, hello, I would like to request you that why not to have some series, because we believe in series.
Yes. So cancer is with him. Dr. Shivsan agreed that once paper is published, he will talk on liver medicine.
I think you have got a very good person along with you, Dr. Jaisri madam.
Yes.
I also have worked with her in my same hospital.
Yes. Simon.
I am Dr. Adar Khedkar. I was pediatrician HOD Department of Pediatrics, J.Y. Patil Medical College, and I have done 55 clinical trials. What I suggest is that we, as Vinay had said, the randomized control trial will be the answer, and all those doubting Thomases will start using this stuff if one RCT shows the results. So I feel I recommend it strongly to Hanundrao sir and congratulate you for doing wonderful work. And this is as Suresh Rao has said that it is a work for the humanity. And one of my closest friends, our friend, is having prostate cancer and he is on these medicines and is doing well. So thank you, sir.
So I think we are already doing that because I went to Tata Cancer, that's where I did my PhD with Pawan Singh. So we are actually getting involved into clinical trials.
Tata Cancer will. So actor Sanjay Dutt is on BVG medications for the last 5 years. And he now publicly acknowledges that when Nitin Gadkari told me to give our medicine to him, and he was a little bit skeptical. So this is scientific, this is for journal. एक मिनटाचा व्हिडिओ
हनुमंतराव गायकवाड़ इन्होंने इनकी बीवीजी कंपनी के द्वारा पार्किंसन और कैंसर इसके ऊपर जो दवाई तैयार की है अनेक लोगों को देने का काम मैंने किया। विशेष रूप से पार्किंसन की दवाई करीब 300 लोगों को मैंने यहाँ नागपुर में अन्य जगह पर दी, जिसमें पार्किंसन की कोई दवाई नहीं ऐसा डॉक्टर बोलते हैं, पर इस दवाई का बहुत अच्छा परिणाम आया। अनेक लोगों को बहुत आराम पड़ा, कैंसर रोगियों को अच्छा अनुभव हुआ। संजय दत्त जो फिल्म स्टार है, उन्होंने मेरे कहने पर ही उनसे दवाई ली और उनको भी काफी बड़े प्रमाण में कैंसर और बाकी तकलीफों के लिए बहुत अच्छा फायदा है। मुझे लगता है कि ये जो रिसर्च करके जो दवाई है उन्होंने तैयार की है वो बहुत अच्छी है। जरूर जो विशेष रूप से इस प्रकार की रोगों का सामना कर रहे हैं जरूर इसका उपयोग करके देखें, ये विश्वास के साथ मैं करें। धन्यवाद। हनुमंतराव गायकवाड़।
संजय नमस्कार। औषधी मॅडम सर, त्यावेळेला त्यांना ऑनेस्टली मी त्यांना स्वतः द्यायला गेलो होतो औषध आणि ते 2 रुपयाच्या खाण्यावर हात टाकून मला दवाखान्यातून सोडलं। 2 रुपयाचा, आता ते 6 महिन्याचा स्टॉक ठेवला होता औषधाचा आणि आता जे नवीन पिक्चर 14 तास लक्ष दिलं।
नमस्कार, मैं संजय दत्त और मैं ये कहना चाहता हूँ कि डॉक्टर गायकवाड़ की दवा मैं अभी 3 साल से खा रहा हूँ और गडकरी साहब ने मुझे बोला कि बहुत अच्छी दवा है और मैं हर चीज़ की दवा खा रहा हूँ गायकवाड़ साहब की और बहुत बहुत मुझे फ़र्क पड़ा है, बहुत अच्छा लगा है। ये लीवर की दवा है जो मैं खाता हूँ रोज़, ये कैंसर के लिए दिया था उन्होंने, वो भी लेता हूँ और ये हार्ट के लिए ये भी लेता हूँ। तो डॉक्टर साहब आपका बहुत बहुत धन्यवाद, थैंक यू वेरी मच सर। और इससे बहुत मुझे मदद हुई है और थैंक यू, आपने मुझे ये दवाई दी, थैंक यू। और बहुत अच्छी दवा है, थैंक यू।
So you can use it for prevention of cancer?
I should say. रोज 5 जर घेतलं ज्या घरात हिस्ट्री आहे तर ती वाढ होते। मला मी काही डॉक्टर नाही, माझा भाऊ डॉक्टर आहे, डॉक्टर इंजिनियर आहे। मी यांचं सगळं ऐकून यांनी सेल ग्रोथ थांबते, म्हणजे जवळपास जीवनची 2 सायकल जे असतात 94% पर्यंत अरेस्ट होतात, सो ग्रोथ थांबते आणि ही ऍक्शन 7 दिवसात होते। म्हणजे लास्ट इयरच्या पेशंटला मी भारताच्या सरन्यायाधीशांच्या घरी होतो 4 वर्षापूर्वी आणि तिथं सद्गुरू जगी वासुदेव होते आणि मी कुठल्याही अनुभवाशिवाय बोलत नाही। मी तोपर्यंत किमान 100 पेशंट झाले होते आणि मी पेशंट स्वतः बोलतो तुम्हाला काय फरक पडला, तो पेशंट सांगतो मला भूक लागायला लागली, माझं वजन कमी झालं, वजन वाढायला लागलं, मला झोप लागायला लागली, स्पोर्ट सकाळी 7 दिवस व्हायला लागलं, म्हणजे माझं सगळं आयुष्य नॉर्मल झालं। मी त्यांना म्हटलं की सर माझं असं औषध आहे आणि त्याने फायदा होतो। मला म्हटलं मेरी भी दवा बोली मेरे पास 14-15 फॉरेनर्स है, लास्ट स्टेज में बहुत पेन है, तुम्हारी दवा से क्या फर्क हुआ? 15 दिन के बाद बात करें आणि सर सांगायला खरं आनंद आहे की मी दुसऱ्या दिवशी औषध पाठवलं आणि 12व्या दिवशी त्यांच्या मामा म्हणून आहेत ज्या हेल्थच्या प्रमुख आहेत, त्यांचा मला फोन आला की सगळ्या पेशंटना वेगवेगळे कॅन्सर होते, प्रोस्टेट, पॅन्क्रियाटिक, कोरोन, ब्रेस्ट आणि सगळ्यांचा पेन कमी झाला। सर तुम्ही की हनुतला असं त्या पुढे गेला पूर्वीशी त्यावेळेला सगळे पेटेंट वगैरे फाईल झाले होते. साहेबांचं रिसर्च चालू होतं म्हणजे आपल्याला सगळ्या कम्युनिटीला सांगायचं होतं की आपल्याला बाकीच्या कंपन्यांसारखं सांगून उपयोग नाही, आपल्याला सायंटिफिकली गेलं पाहिजे आणि ते काम आम्ही करतो. सो असं चालू आहे, जर्नी चालूच आहे. त्या जर्नीला तुमच्या सगळ्यांचे आशीर्वाद मिळावे. तुमच्या माहितीतले लास्ट स्टेजचे फर्स्ट स्टेजचे कुठलेही पेशंट तुम्ही रेफर आणि आपण हे महिन्याचं असं 5,000 रुपये देतो. माझा अनुभव आहे की 8 ते 10 महिन्यामध्ये त्या पेशंटला प्रचंड फरक पडतो. म्हणजे एक महिन्याभरात सर बोलले सर ज्या माणसाला बेडवरून उठता येत नाही तो महिन्याभरात चालला. प्रबोध हळदे म्हणून माझे एक मित्र त्यांनी मला व्हिडिओ म्हणून नाव घेतलं. आपल्या ह्याचे पॅराशूटचे जे मरेटूचे त्याचं स्वतःचं ही चेअरमनचा असिस्टंट एक्झिक्युटिव्ह असिस्टंट टू चेअरमन मरेटू. त्यांच्या आईला कॅन्सर झाला होता आणि त्यांनी मुंबईत ट्रीटमेंट घेतली मोठ्या मोठ्या हॉस्पिटलमध्ये आणि सांगितले की 15-20 दिवस राहिले आणि ते गडकरी साहेबांच्याकडे गेले. गडकरींना सांगितले की असं असं झालं. गडकरी साहेब सांगितले की हंड्रेडच्या औषध नाही घेऊन 15-20 दिवस राहिले काय करायचं त्यामुळे तरी ट्राय कर. मला तिथूनच फोन केला मी बसमध्ये औषध टाकली पुण्यात नागपूरला पोचली दुसऱ्या दिवशी चालू केली. त्याचे 7 महिने चांगली राहिली. 3 आठवड्यात घरातली सगळी कामं चालू केली. असे अनेक अनुभव माझ्याकडे की आता औषध कसं काम करते मला माहित होतं. तुम्ही जे मगाशी म्हटलं की पेशंटला फरक पडला पाहिजे कसं काम करते हे फाइंड आउट करायचं चालूच आहे. पण आता हे पण कळलं की मल्टिपल कॅन्सर होते ते कसं कसं काम करतं. सो म्हणून आज आपण जो 8 तास घेतला की तुमच्या सगळ्यांचे आशीर्वाद मिळावे. तुम्ही प्रत्येक जण रोज 50-100 लोकांना भेटतात आणि हे पैसे नाही करत. माझं ध्येय की ह्युमॅनिटी आहे माणुसकीसाठी काम करायचं 10 कोटी लोकांचे असे बदल करायला मदत करायची. या सगळ्या मिशनला तुमचे आशीर्वाद आहेत हे सगळं सगळ्यांना.
I'm Dr. Shailaja Mani. I'm professor at the Department of Pediatrics, D.Y. Patil Medical College, and I'm trying to continue the legacy of Dr. Agarkar sir and working on many ICMR and John Hopkins projects as well. First of all, heartiest congratulations, Mani sir, and backwards our entire team of BVG and team which is working at US. I want to ask 2-3 things. Do you think that we should continue with adjuvant or only?
I think both.
म्हणूनच घ्या आणि मग जरा अजून रिसर्च झालं की मग आपण
Yeah, that's what. RCTs and that will give a lot of answers to many questions. And I want to ask whether phase 3 clinical trials are conducted anywhere. Want to? Okay. Then we like to join the hands, particularly in cancers and Parkinson, and particularly because I am a pediatrician and we are having one unit of pediatric cancers in DY also. We like to join for the further studies and for excellent cause of the humanity as well. Thank you.
Ma'am, being a consultant with BVG since last 3 years, I am keenly observing the patients very in detail. So our way of practice right now is when patient come to us, we never tell them to stop their ongoing treatment. First of all, we never claim that you will have a 100% cure, but we teach them that your life quality will be improved. You will have this medicine as an adjuvant that will help you to have your quality improved. Automatically your life expectancy will be better. So this way we are practicing right now. We always teach them to take the medicine together. And regarding the Parkinson also, we have very good medications. That was an accidental finding and a feedback from the patient. When he takes some medications for the immunomodulator, they have some feedback that their tremors have been reduced. And after that, then we start working on the Parkinson's also. Regarding the RCTs, we are in touch with Tata Memorial for different conducting the trials. Once we get permission, we will be conducting the trials at Tata Memorial. Thank you so
With
much.
HMSC permission.
Yeah. Any other questions anyone want to? So thank you so much for patient listening. I would like to extend our sincere gratitude to Dr. Shrikant Manesar for today's outstanding presentation and incredible insights. So concluding today's session, on behalf of BVG Life Sciences, we express our sincere gratitude to our esteemed guests for sharing their invaluable insights, expertise, and research findings. This reflects years of dedication, and certainly it will help for clinical development aimed at improving patients' outcomes. I extend our deepest appreciation to all the attending respectable doctors for their active participation throughout the session. Looking forward for future collaborations in interest of patient care for the better outcome. Thank you so much, everyone.